Hi science friends, let's review just a little bit from yesterday. So the first one, without using your notes, name the 4 major processes of cellular respiration and where they occur in the cell. So number 1, you should say glycolysis. Hopefully you knew that one to start out with. And that happens in the cytoplasm. Both in prokaryotes and eukaryotes it occurs in the cytoplasm. The second step is the kind of the stepping stone step called pyruvate oxidation. And that happens in a eukaryotic cell in the mitochondria. In a prokaryotic cell, that would occur in the cytoplasm. In the third step, that is the citric acid or the Kreb cycle. And that also happens in the mitochondria or in a eukaryotic cell that doesn't have a mitochondria in a cytoplasm. Sorry, that was a little messy. And then finally, the last step is the electron transport chain / chemiosmosis, which is an example of oxidative phosphorylation. And that happens in the mitochondrial membrane, specifically the inner membrane of the mitochondria, or in the prokaryotic cell in the cell membrane. Okay? And so that's the 4 steps. You should know those. Okay? The next one, explain the difference between oxidative and substrate level phosphorylation. Okay? So the first 3 represent substrate level phosphorylation. Oxidative phosphorylation only occurs in the electron transport chain. But more importantly than that, what's the difference in them? And the difference is where the phosphate comes from. So in substrate level phosphorylation, the phosphate is stolen from another substrate. It's popped off of another molecule and added to ADP to make ATP. That's what phosphorylation is, putting a phosphate on something else. And here we're talking about putting it on ADP to make ATP. And that's really the goal of cellular respiration, to make ATP from glucose. Okay? Now that was substrate level phosphorylation. It's stolen from a substrate. In oxidative phosphorylation, the phosphate group comes from inorganic phosphate that's present in the cell. Okay? And so it's not really stolen from somewhere else, but it just comes from inorganic phosphate available in the cell. And again, that's in the electron transport chain. Okay? So today we are going to talk about, I'm not sure, my headings are wrong on this PowerPoint. So change the headings in your notes. It should say pyruvate oxidation. That's what we are going to talk about today. Pyruvate oxidation. And I think it's wrong maybe, okay, I guess that's the only place it's wrong. I thought it was wrong somewhere else too. But change that heading to there. And so this is the second step in the process. So let's go ahead and get our paper out and kind of draw to review from yesterday. So step 1, we had glycolysis. And glycolysis, remember, is the breakdown of glucose. Okay, so glucose is C6H12O6. And in the process of glycolysis, we break it down into 2 3-carbon molecules called pyruvate. Okay, this happens in the cytoplasm, as we mentioned earlier. And when we break this molecule down, we break that bond, we release energy. There's 2 net ATP produced. When we break that bond, we also release electrons. And those electrons get picked up by NAD plus to form NADH, which takes the electrons to the electron transport chain. Okay? So now we have these 3 carbon molecules of pyruvate, okay? We are only going to really follow one of the pyruvates through, but realize that there's 2 pyruvates that are made, and so both of them are going to have this same fate, okay? So in the next step, step 2 is pyruvate oxidation. So it's being oxidized, and so that's a loss of electrons. So pyruvate's being broken down. And so pyruvate is converted to a molecule called acetyl-CoA. Okay? Converted to a molecule called acetyl-CoA. Now this happens in the mitochondria. And so how did we get from the cytoplasm to the mitochondria? Well, we got there by this guy, a little transport protein. Okay, so there's a transport protein that carries the pyruvate from the cytoplasm to the mitochondria. All right, and so transport proteins, they can be, most of the time are going to require energy, okay, to be able to move that in. And so that's why this process doesn't really net any energy. All right, so it's active transport. Okay, it requires energy, it's active transport, and pyruvate gets converted to acetyl-CoA. So, yesterday I said something that was a little bit wrong. I told you that pyruvate, in the conversion to acetyl-CoA, lost an oxygen. And while that's partly true, what it really loses is carbon dioxide. Okay, carbon dioxide is really what's being lost here. So if you compare the structure of pyruvate to acetyl-CoA, you see they both have a CH3, a methyl group. They both have this C double bond O, which is a carbonyl group. And then pyruvate also has this CO2, which it loses, and you see that here. If you think back to our equation for cellular respiration, C6H12O6 plus O2 yields CO2 plus H2O. Carbon dioxide is one of the products of cellular respiration, and here's where it comes from. Okay, now it's also made in the Kreb cycle, but this is the first place that carbon dioxide is made. All right, so 1 molecule of CO2 is released here. We also have an addition of coenzyme A, which you see here in the structure of acetyl-CoA. Now, coenzyme A is a coenzyme. We've talked about those before. And so it's a derivative of vitamin B. And so that's why vitamins are important in our bodies and to get in our diets, because they help make up enzymes that keep these metabolic processes happening. Now, vitamin E is in a lot of foods. It's in a lot of whole grains. It's in meats. It's in vegetables. It's in fruit. It's in a lot of things. And so that's not usually one we really have to focus or try real hard to make sure we get, but it's in a lot of foods. Okay? So again, this is oxidation. Loss is oxidation, so losing electrons. So when this pyruvate is broken down, it loses electrons. Well, guess where they're going? They are going to be picked up by our electron carrier, NAD, to form NADH. Where do you think NADH is going to take them? To the electron transport chain. Okay? Hopefully you're seeing some patterns here. And so the goal of this step, pyruvate oxidation, is to convert pyruvate, which came from that glucose. Remember, we really have 2 of them that we're having to convert and do all of this to acetyl-CoA, because acetyl-CoA is going to enter the next step, which is our Krebs or citric acid cycle. All right, and in this process, pyruvate oxidation, we release CO2 as a byproduct, and we also release NADH, which goes to the electron transport chain. This part does not make any energy. No energy is made here. So you don't ever see it referred to as substrate level or oxidative phosphorylation because there's no energy that comes from this phase. All right. So that's pyruvate oxidation. Now, this part of the diagram here is still showing pyruvate oxidation. We've got our pyruvate. It's those 3 carbon molecules. We've released CO2. We've lost some electrons that were picked up by NADH. We added in that coenzyme A, but ultimately what we converted to was acetyl-CoA. And that acetyl-CoA is going to enter the citric acid cycle. When acetyl-CoA enters the citric acid cycle, it joins with a molecule called oxaloacetate. Okay? And oxaloacetate plus acetyl-CoA together make a molecule called citrate. Citrate, and this is where we get the name the citric acid cycle, or the Kreb cycle from Hans Kreb that discovered it in like the 1930s. Okay, so that's where you can also hear citric acid cycle. And so the next steps in the citric acid cycle, ultimately the goal is to convert citrate back to oxaloacetate so it can take in another acetyl-CoA, because it's a cycle. We have to keep this stuff moving, right? And so in the process of the citric acid cycle, 2 more carbon dioxides are released. Again, this is more redox reactions. We've got oxidation and reduction continuing to go on, breaking bonds, breaking things down, moving molecules, all those types of things. It's a series of chemical reactions. But we release 2 CO2. We make 3 NADH. Okay, these numbers are important. 2 CO2, 3 NADH. We make 1 ATP and 1 FADH2. Now, what is FADH2? FAD and FADH2 are another set of electron carriers. Okay, FAD picks up the electrons and becomes FADH2. Flavin, adenine, I don't know what it's called. Anyway, it's an electron carrier. And so it is also going to take those electrons to the electron transport chain. Now, NADH is the better guy. He makes more energy in the electron transport chain, and we'll talk about why that happens later. But FADH2 is important to this process too. Okay? So these numbers are representative, these 2 and 3 and 1 and 1. These numbers are representative for 1 pyruvate molecule. Okay? But remember, you have 2 pyruvates for every 1 glucose. Okay? So if we're talking about per 1 glucose, we're going to make 4 CO2s, and we're going to make 6 NADHs, and we're going to make 2 ATPs and 2 FADH2s. Okay? But usually, for simplicity's sake, we talk about per turn of the cycle, and that's per pyruvate or acetyl-CoA molecule. All right? Here's where I had messed that up. From pyruvate oxidation. I knew I had another spot in my notes that was messed up. Acetyl-CoA from pyruvate oxidation, so make sure to change that in your notes, enters the citric acid cycle and combines with oxaloacetate to form citrate. And citrate is the ionized form of citric acid. Okay. So, and that's what we just talked about. That's just expressing it in word form here. All right. So this last slide that I want to talk about today is the whole picture, the whole diagram. So I showed you the whole diagram for glycolysis yesterday, and I said, remember, we're not learning all of that. We're just trying to see the big picture. So here's the big picture of this one. Acetyl-CoA comes in from pyruvate oxidation. It joins with oxaloacetate to form citrate. Okay? And then these remainder of the steps 2 through 8, are trying to get back to oxaloacetate so you can accept the next acetyl-CoA. In this process, we make 1 NADH, 2 NADH, 3 NADH. Okay, we make 1, 2 CO2. Remember, carbon dioxide again is a product of cellular respiration. We make 1 ATP, and we make 1 FADH2 per turn of the citric acid cycle, per pyruvate molecule, and you have 2 of those, which make 2 acetyl-CoAs per glucose. Okay, so let's go back to our paper and let's draw this in here. So pyruvate is converted to acetyl-CoA. In this process, we have to add in coenzyme A. We're going to release 1 CO2, okay? And we are going to release some electrons, which get picked up by NAD to form NADH, which go to the electron transport chain. Okay? And then this acetyl-CoA, scroll down here a little bit. Which enters in the Krebs or the citric acid cycle. And in this process, we release 2 CO2, we release 1 FADH2, which is that electron carrier, we release 1 ATP, and we release 3 NADH. Where you draw these on the cycle doesn't really matter to me. I don't care. Okay? This acetyl-CoA comes in, binds with oxaloacetate to form citrate. Okay? And again, this is happening also in the mitochondria. And this is step 3. Okay? That acetyl-CoA that was made from the pyruvate oxidation. So I hope that helps to clear up that part. Tomorrow we'll talk about, or Monday I guess rather, we'll talk about the last step, which is the electron transport chain. Okay? If you have any questions, let me know. And if not, then you'll have a great day and a great weekend.